Hi, everyone. It's my pleasure to introduce Dr. Dennis Selkoe, who is the Vincent and Stella Coates Professor of Neurologic Diseases at Harvard Medical School and also co-director of the Ann Romney Center for Neurologic Diseases at the Brigham and Women's Hospital in Boston. With us today is Audrey McNiff. She's a significant supporter to the Ann Romney Center. Stand up a moment, Audrey. Thank you. Professor Selkoe's research on amyloid B protein was pioneering in Alzheimer's disease and led to the amyloid hypothesis. That in turn led directly to the first effective therapies for Alzheimer's disease that most of us have heard or read about in the last year or two. His lab is also doing and has done important work on alpha-nucleoside, which is a key protein in Parkinson's disease, and I suspect we're going to be hearing shortly about new therapeutic advances based on that. Dr. Selkoe is a fellow of the American Academy of Neurology and a member of the National Academy of Sciences. He's a founding scientist. He was a founding scientist of Athena Neurosciences and is now a founding director of the Prothena Biosciences. It's my privilege to welcome Professor Dennis Selkoe.
Thank you. Pleasure.
Sir.
So I'm delighted to be here. I'm familiar with this territory, and I want to thank my neighbor, Audrey, and who introduced me to Ken. We had a wonderful dinner, one of several at Audrey and Tim's house, and I'm really happy to be with you all, and I'm very grateful for the invitation. My goal today is to share with all of you what's new about this difficult, really very sad disorder we call Alzheimer's disease. And I'd like to leave a fair amount of time to answer your questions at the end of all this. Okay, we're going to bring in more guests, which is great. So I work at what's now called Mass General Brigham. Originally, I worked at Brigham and Women's Hospital, so I'm a professor at Harvard Medical School, and we do a lot of research on neurologic disorders. My colleague Howard Weiner is a world authority on multiple sclerosis, and we work on other diseases like ALS and brain cancer, etc. But today I'm going to talk to you about Alzheimer's disease, and let's see if we can go back to the slides. If I push this, let's see if you can see that. Okay, can you see that slide? Good. So what I'm going to share with you today for about 25, 30 minutes, and then we'll have an open discussion, is the quite remarkable progress that's been made worldwide in understanding the cause and the mechanism of Alzheimer's. And you wouldn't know this by reading the New York Times or the Wall Street Journal or any regular newspaper, because they often haven't gotten it right. They haven't understood what has happened in the scientific field. So it's been 40 years. I've actually worked even longer than that. I've seen hundreds and hundreds of patients with Alzheimer's, and we've figured out what we think is a way to slow it down or treat it. And you'll have different things you've heard in the media or from friends or colleagues or doctors that I hope you'll ask me about, because there is a fair amount of confusion about how Alzheimer's works and what to do about it. So here's some statistics about what we know about the personal and societal impact of Alzheimer's. Can you see the slide all right? So we know that it's a common chronic, ultimately fatal neurologic disorder and affects some 65 million individuals worldwide. As such, it's the most common cause of dementia and death from dementia in the US. And it's marked by the accumulation in the brain of the amyloid beta protein that I'm going to talk about quite a bit, and another protein called the tau protein. These are Greek letters, beta and tau, and that's how we call the abnormal proteins. And Alzheimer's is a huge burden, obviously, on a personal level, as so many of you have experienced, sadly, but it's also a huge burden for our health system here and around the world. So every 65 seconds, someone will develop Alzheimer's in the United States. And we're very concerned about stopping that. And we've now gotten, on July 6, 2023, a red letter day in the study of Alzheimer's, the first FDA-approved drugs that slow down Alzheimer's. And we want to make those widely available. The lead drug has a name. It's called Leqembi. And again, I'll be happy to answer questions after this if you don't catch some of my words. But Leqembi, that I'll talk about, and other drugs, is now approved in 50 countries. And I'm sure that folks in the room may not be aware of that there was actually an Alzheimer drug, and it could actually work for prevention. That's why in my title here I mentioned in question mark, could this be a prevention? And I think it will turn out to be. The cost of this disorder in the U.S. alone is about $400 billion a year. And this is a universal scourge. There's thought to be maybe about 350 million people in the world that have this process underway. So I'm going to share with you some of the ideas that we've come up with across the globe. There have been scientists in many countries, hundreds of labs, hundreds of clinics that have studied this disease to advantage. We know the concept of senile dementia, and I'm going to define this further. The lay public thinks of this as senility, and we've all heard that, and the medical term is senile dementia, which means progressive mental failure after age 65. Now, 65 is the arbitrary Social Security Administration rule about when folks tend to retire. And it comes actually back from Germany, from Bismarck's time. So it has no biologic significance, but nonetheless, we have divided pre-senile dementia, early dementia, from senile dementia at age 65. In real life, it's a continuum. Alzheimer's is very rare in the 30s and 40s, but it actually appears then, and then it rises with time, with age, and it's really common in the 80s and 90s, and even one can become 100 and develop Alzheimer's for the first time. So this is what we call pre-senile dementia before 65, and then senile dementia is the common term that people refer to as senility. You see in the second bullet point here that Alzheimer's is the most common of more than 20 causes. And don't ask me to name all 20 causes. I can probably tick them off, but it might take me a couple of hours. But there are many other causes. There are conditions such as Lewy body dementia, which is a type of dementia, something called frontotemporal dementia, something called vascular dementia, also known as multi-infarct dementia. And today I'm going to focus on the most common dementia, which has the name Alzheimer's disease. But there are other dementias, and the medicine that I'm going to describe to you wouldn't work for those other disorders. It only would work for this condition we call Alzheimer's. And that's the rule in biomedicine. You need to figure out the cause of a disease and then interrupt it with great specificity. You can't just treat Alzheimer's, frontotemporal dementia, and vascular dementia with vitamins or with some kind of food supplement. That might help many different functions in the brain, but it won't specifically nip Alzheimer's in the bud. And so we need to understand what is the biology behind this disorder. So you know that Alzheimer's begins insidiously, very slowly, usually with decreased memory and confusion, disorientation, and progresses slowly over 5 to 20 years. And it is fatal, ultimately. So people who have Alzheimer's versus those who do not at 75 or 80, those who have it will die sooner by 1/3 to 2/3 sooner and less life expectancy. I want to show you this slide, and I warn you, and I'll take you through it as tenderly as I can, as carefully as I can, that there's a little bit of science in what I'm going to share with you. And I'll ask you to bear with it, and I'll be happy to explain it later. But to figure out how to prevent Alzheimer's, which is what everyone in the room would like, we needed to figure out what causes it in the first place. And that came from studying what happened in the brain. And this image is a slide of a little small area of brain, area called the amygdala, in a patient that I followed during life. So the image I made, I took this photograph when we took the brain out after someone died with Alzheimer's. The gentleman was 70 years old. He had a mother who had Alzheimer's before him, and we studied his brain tissue to find out what was going on. And one way we studied it is to stain the brain with a special stain. This is the same stain that this fellow Alois Alzheimer used in 1906. He was a renowned psychiatrist in Munich and also worked in Frankfurt, and he had the first patient with what's called Alzheimer's. He defined the disease. And he pointed out these two round deposits you see here that are orange colored. I think I can you see me move my little cursor here? Does it show up? Yeah. And these are amyloid deposits or amyloid plaques surrounded by abnormal axons and dendrites, the connection of nerve cells. And adjacent are these dark staining nerve cells. These are neurofibrillary tangles. And on the top, you see amyloid plaques. Those are the round items made up of a protein we call amyloid beta, and neurofibrillary tangles that are composed of the tau protein. So this is something we discovered. My lab was involved in the beginning of this in the 1980s. A man called George Glenner at the University of California, San Diego, first described the amyloid beta protein and named it as such because it has certain properties. So I'll ask you to remember plaques in the brain called amyloid plaques and tangles in the brain called neurofibrillary tangles, two different proteins. A beta is one, tau is the other. And I'm going to tell you how they come together and what we're doing about it. We now have treatments that remove both of these from the brain, which is just what we would like to do. Another part of these plaques is what you see here. So you see an inflammatory process around these amyloid plaques. These are inflammatory cells that get angry, get activated. And so these are angry-looking microglia, a certain kind of inflammatory cell in the brain. And here you see a ring of brown cells surrounding this amyloid plaque. These are called astrocytes. So I'll ask you to remember that plaques have inflammation in the brain. This inflammation won't be helped by Motrin or Advil or even prednisone or steroids. It's a special inflammation that happens in Alzheimer's disease. Now, how did we get here? How did we figure out as a community what Alzheimer's is all about? So we studied the prevalence of the disease, and you can see here, I'll go backwards and then I'll go forwards, and you can see this graph of how Alzheimer's rises in prevalence around the world with age. You can see age on the bottom. And what we discovered was the following. If we graphed the number of plaques that people had when they died of various reasons, that also followed a trajectory going up with age, that plaques of amyloid in the brain. And there was a ability shortly in the 1970s or 1990s to image these plaques by something called PET scanning, positron emission tomography scanning. And so you see here, I can go backwards, this is just studying the plaques by examining the brains of people who've died, and this is studying them in real life, the dark blue dots, by an image. And we can do that now, and we're doing it in all of our patients to whom we want to offer this breakthrough treatment that FDA approved. And there's a 15-year interval, as you can see, between the accrual of these nasty plaques in the brain and the development of symptoms of Alzheimer's. So that 15-year interval gives us a great opportunity. That's where we want to intervene, ultimately to prevent all of us from getting the disease. You won't need to read all this. This is a model or a theory behind Alzheimer's that Ken nicely referred to. The amyloid hypothesis that I published in 1991, colleague of mine, John Hardy, with whom I just spoke a couple of days ago, published a version of this also in 1992, and we were there at the same moment in time proposing a controversial theory that the plaques in the brain were fundamentally the cause of the disease. And if we could get rid of them or prevent them, that would be good news. So a complex cascade, it isn't actually fully correct the way I wrote it back in 1991 in April, but it has the elements of future success. Now, this is the hypothesis that we've been following. And it's been controversial. The papers have said, well, a lot of doctors don't think this is right, and there's a lot of debate about all this. I'm going to read it to you. Alzheimer's disease could be a syndrome caused by an imbalance between the production and the clearance of this amyloid beta protein that leads to its gradual accumulation and aggregation in brain regions serving memory and thinking, cognition. And the imbalance, we thought, could be caused by different genetic factors. You could inherit the disease. We know that some of you in the room probably have a history of someone in the family who might have had Alzheimer's. And there could be environmental factors. And we don't know as much about those, but there's some clues. So this was a hypothesis, debatable, controversial, but it's turned out largely to be correct. Now I'm going to give you this image, which is quite striking. It's a picture in the brain of these amyloid plaques or amyloid deposits. They're in brown here, and the lower part of the image is white. It doesn't have plaques. And that is the white matter in the brain. If you've ever heard about the brain having gray matter and white matter, how many of you have ever heard those terms, gray matter and white matter? Right. So the white matter doesn't get any plaques. The gray matter, which is the rest of the slide, gets lots of plaques. Now, if I told you that this image was taken from a patient that I followed during life who died at age 12 with all these plaques in his brain, you would be absolutely shocked. And that was a huge clue to us. How could someone age 12 have amyloid deposits like we see in 80-year-olds with Alzheimer's? And the answer is this is an unusual condition, but not unheard of. You'll know the name when I give it to you. That leads to a very premature buildup of amyloid in the brain, of early form of Alzheimer's. Can you imagine what diagnosis could give these plaques in the brain that look like Alzheimer's at age 12?
Progeria?
Progeria is a very good choice. That's a premature genetic form of aging. That happens not to be the case. Progeria doesn't bring the amyloid plaques. I don't know if there are any ringers in the audience who know what condition happens to humans that produces this. And if there aren't, any other guesses? So the answer is Down syndrome, also known as trisomy 21. So Down syndrome is something we're familiar with in society. We all have encountered individuals, some of them may be in our family, that have Down syndrome. And Down syndrome, 100% of the time, leads the patient to develop amyloid plaques and later neurofibrillary tangles in the brain like you see in Alzheimer's. So an enormous clue when that was discovered in the 1970s at Duke University. And it turns out that the gene that makes the amyloid protein that I'm showing here, that makes all these brown deposits, happens to be on chromosome number 21. And therefore, people who have 3 copies of chromosome 21 instead of the normal 2 copies would develop Alzheimer's disease prematurely. So that leads to, and there's the answer to my quiz, 12-year-old boy with Down syndrome. And that brings us to the genetics of Alzheimer's. So you have to choose your parents very carefully. It's very important to do that. And if there's any one message I want to leave you with, that's it. And I didn't get around to doing that myself, but I'm sure that we can get to that. And when we looked at people who had a family history of Alzheimer's, the first mutation in the DNA we found was on that first line, an APP mutation that you see right here. And the arrow is up. The amount of A beta or amyloid beta in the brain increased when you had a mutation in chromosome 21. The gene APP is on chromosome 21, and if you have trisomy 21 or Downs, you have an extra copy. If you have one of these mutations, you could be not like a Downs patient, just a conventional individual, what we call wild-type people. That's what you all are, just wild-type, natural, and yet have a single-point mutation in the APP gene that gives you Alzheimer's. And then these others that I won't go over in detail are other genetic forms of Alzheimer's. ApoE4, which is a very important gene, presenilin 1, presenilin 2, which we discovered, is the enzyme that makes the amyloid. And the point of this slide is that all of these Alzheimer causative mutations raise the amyloid level. The arrows point up, and that's bad news. We don't want to have these. Now, a lot of folks develop Alzheimer's without any of these genes being abnormal or altered, even without the ApoE4. But especially these 4 genes would lead to an aggressive form of Alzheimer's. This is an image from work that we did in Nature in 1999 showing an enzyme in yellow and red. You don't need to know the details of this. And that enzyme is called presenilin. Now, presenilin should be a name that rings a bell with you. It would cause a presenile form of Alzheimer's. And I mentioned earlier, age 65 is the cutoff between presenile and senile Alzheimer's. So presenilin is an enzyme that leads to premature early Alzheimer's, as early in my experience as a 18-year-old girl in high school who developed Alzheimer's and died at 33. She wasn't my personal patient, but I've read about her, I know about her. I think she's the youngest patient in the world reported with Alzheimer's. So presenilin is the enzyme that makes that little amyloid beta peptide that makes all the plaques. And presenilin has a mutation in this 18-year-old girl. She was born with that, and that mutation led her to make an abnormal form of the amyloid beta protein. So this enzymatic reaction, what you're seeing here is the amyloid parent protein, APP, is in this groove in presenilin, and then it's cut, and the little purple piece that's let go from the membrane is the amyloid beta protein. So let's talk about how this intensive biological research that I've given you a little flavor of panned out over 4 decades to allow the FDA, they were the first in the world, and then the EMA, the European Medicines Agency, and then 50 countries, China, Japan, all over the world, Israel, to approve immunotherapy, antibody therapy. So if you're familiar with the concept of immunotherapy or antibody therapy for cancer, then we're talking about the same kind of thing, but for Alzheimer's disease. So this is not cancer, I don't want to confuse you, but antibody treatment can be used for a number of diseases, rheumatoid arthritis, etc. You can see the advertisements for psoriasis on the news. And in our case, we have immunotherapy against this amyloid protein that shouldn't build up in our brain. We don't want it there. This is the headline from our global network called ALS Forum that we all go to every Monday morning to see what's new in the field. And all of my students in the lab at the Brigham read this to find out what's new. And the big news on October 3rd, 2022, that you see here, was finally a big win on all outcomes for lecanemab in phase 3 top-line results. So ironically, we saw this headline in our communication, but the world hardly noticed that. There wasn't a lot of discussion about that because there was still debate about whether lowering amyloid would help people with Alzheimer's. I'm going to show you a couple of examples briefly of how we showed that the amyloid buildup could lead to secondary problems in the brain. So what we do is we look at a control brain, that is someone who died at age 75 or 85 without Alzheimer's, and it looks like this on our protein gels, that's what you're seeing here, versus a lady who died with Alzheimer's disease at 80, and she has these black lines which represent the amyloid protein in the brain. In these parts here, we can separate a dimer, which is a doublet of 2 A-beta proteins, or the A-beta monomer, which is a single protein. So this is the beauty of biology in a laboratory. You can actually take the brain of someone who died with the disease and isolate the abnormal material from the brain. And what we found when we took this lady's brain extract, and put it on healthy neurons. Well, these are beautiful nerve cells or neurons. That's what neurons look like. Some of you may have seen an image before of a nerve cell looking like this. And all of these rows, all of these columns are normal except the last one. The last one is when we took an Alzheimer saline extract from the lady who died with Alzheimer's and put it on these neurons, and it's this material, fractions 8 and 9, which have doublets of the amyloid beta. So the singlet of the amyloid beta, just the single molecule, didn't hurt these neurons in our experiment. But here on the right, the doublet of amyloid beta was damaging. That doublet is what we call a dimer, two amyloid proteins together. And the analogy I like to make is if you see a tough guy on the corner one night at 9:30 or 10, the one guy alone probably is not going to bother you too much. You walk past and go to your car. But two guys start getting too much testosterone and start getting a little bit difficult. With 3 guys or 4 guys, you really get nervous. And that's the case with this amyloid protein. The one guy, the singlet, isn't a problem. We actually all make it throughout life, and our lab at Brigham discovered that. But doublets and higher are trouble, like the guys at the corner. So this right here, this image is bad news. You can see the neurons don't look so good here compared to here. The abnormality that you're seeing here is in the neuron in another protein called tau that makes up the tangles. So we always wondered, how does the amyloid buildup relate to the tangles of Alzheimer's? That's what Alzheimer described. And we found in this image that you could see abnormal tau. These nerve cells don't look as healthy as these guys here, which is a control. But if we did a genetically engineering trick to decrease the tau in the brain that you see here in black, this is the whole row of tau staining, then the neurons looked fine. And so lowering the tau in the brain kept the amyloid protein from doing its dirty work, suggesting that the lesions that Alzheimer described, amyloid plaques, tau tangles, are related to each other. If the amyloid builds up and you put it onto neurons, you get abnormal tau, and if you knock down the tau through genetic engineering, we wish we could do this in all of us, then you don't get any damage. So that means that even though there's a new antibody that I'll tell you a little bit more about before I close now, that clears the amyloid out of the brain, the tau protein is very important as a mediator of badness, as a fellow traveler, a guy who's going to cause trouble. And that tau protein is now being knocked down, as you see here in these black images here. The tau has been lowered. It's being done in people by Biogen, a pharma company that is giving a tau antisense oligonucleotide, fancy word for a special little fragment of RNA that can lower the amount of tau. So a second way to treat. And this is the way that we treat nowadays. This is treating these nerve cells with an anti-amyloid antibody. There are 2 examples here that you see. And in this experiment we did, 2 of these antibodies, 3D6 and 82E1, worked well. A third antibody, 21F12, didn't work because you want the nerve cells in our experiments to look like this, not like this. And when you add 3D6 or 82E1, you rescue the nerve cells from this damage from the amyloid protein. So that's enough science. I apologize for burdening you with that, but it's really important for you guys and me to get proper treatment for this disease. So towards disease-modifying treatment and the prevention of Alzheimer's disease, how are we doing? Well, we didn't do well for a number of years, which is why the newspapers didn't cover this. There was a lot of controversy. Here are 10 different agents or treatment agents on the left column. This is how they're supposed to work in the brain. And these are the liabilities that each of these possible treatments had. I won't want you to read this off now, but in each case, they failed to actually lower the amyloid. They were advertised, and my patients went into these trials as being given an amyloid-lowering agent. But when we tested it out, none of these 10 did anything. And there's an even longer list that I'm not going to show you. So this is one of my favorite slides when I teach at Harvard Medical School or tell scientists about this story. The reason we failed and that everyone said, well, this amyloid lowering doesn't work, it's nonsense, it doesn't work at all, was that all these trials didn't actually lower the amyloid protein. Sometimes they didn't even try to measure it. Sometimes they included patients in their trials that didn't have Alzheimer's by mistake. So we got around all that. We figured that out, and now we are doing trials right. A colleague of mine, Dale Schenk, at a company that I helped to found, Athena, did this first experiment. This was a remarkable original observation. Dale Schenk treated mice that develop Alzheimer's disease. We engineered those mice with saline. And saline treatment, salt water, didn't help these mice. They got lots of brown amyloid plaques in their brain. But when he gave the amyloid protein itself to the mice by immunizing, the amyloid beta went away in C and D. So this was the concept. You could use an antibody or a vaccine to cure or treat a mouse. Now, in biomedicine, we've cured a lot of diseases in mice. And the question is whether we can do it in you and me. And it turns out that that insight, that's how basic science works, was translated into treatments. And here you see the first remarkable example. This was an antibody made according to this fellow Dale Schenk's concept and based on my hypothesis that amyloid caused Alzheimer's. And the antibody had this name aducanumab. MAB at the end of these strange names means monoclonal antibody. So this was a monoclonal antibody to the little amyloid protein. And here you see what happened between baseline at the beginning of the trial and 1 year of treatment. On a placebo, the red signal, which is the amyloid in the brain, didn't go down. But when you got to 3 milligrams per kilogram body weight per month, or 6 milligrams or 10 milligrams per kilogram body weight per month, you see what you saw here, this remarkable removal of the red signal being a way of imaging the amyloid deposits in a person's brain. So these are real people. This is one person, this is another person, this is a third person, this is a fourth person having Alzheimer's disease, and we cleared the amyloid in this trial. And so that was wonderful news, but it was very difficult to then show that this helped thinking. But when another antibody, not that first one, but one called lecanemab, again, MAB, monoclonal antibody, was used in early stages of Alzheimer's disease, what we call mild cognitive impairment, or MCI, there were very good results. This was a New England Journal of Medicine article, and the results looked like this. This was a cognitive test that we gave people over 18 months, and we gave them either placebo, which is shown in blue, or lecanemab in yellow. And you can see the separation of the lines. The patient still went downhill, but not as much on Leqembi or lecanemab as on placebo. This was their brain level of amyloid after these 18 months of treatment. There was a dramatic drop here in yellow in the amyloid level in their brain, whereas the placebo, of course, allowed the amyloid to stay. And then three other cognitive tests. And what we noticed was that over treatment of 18 months, these lines right here separated. That is, the placebo would get worse and worse, the patient with Alzheimer's not as much. We've now developed a new antibody, we call it B28, thanks to philanthropic support at the Ann Romney Center, that does this. It clears these amyloid plaques, which are red dots, compared to antibodies that don't do that, don't work in control. And then finally, in green versus red, our new antibody, B28, has about 3 to 5-fold more ability to bind up the amyloid protein in Alzheimer patients' brains as with lecanemab or Leqembi. So let me summarize what I've told you. So as of today, I've made the argument that amyloid, and especially A beta dimers and these little monomers coupled together to each other called oligomers, fumers, facilitate tau alteration and spread in the brain and stimulate glial inflammation. So that's bad news. Lowering the amyloid levels leads to lower tau level, that is, less tangles in the brain. So that's good news. So A-beta antibodies, now approved in 50 countries, modify the defining lesions of Alzheimer's. And amyloid lowering in other brain diseases, there are cardiac diseases that Ken would know a lot about compared to me. It's something called transthyretin amyloidosis. That has worked. Antibodies to that and other treatments have lowered the heart amyloid, different amyloid, and that has helped patients with that. So while many factors contribute to Alzheimer's disease genesis, this amyloid beta peptide has emerged as the most scientifically compelling therapeutic target and has now been validated in the clinic, that is, in clinical trials. So at Mass General Brigham, for example, we've now treated almost 900 people with typical mild Alzheimer's disease with this agent Leqembi, or lecanemab, and a sister therapy from Eli Lilly Company that's called donanemab. And they really seem to work. So finally, there's some questions remaining, and then I'll take your questions about all this. Following our initial success, can these anti-amyloid doses that we give people be lowered by 50% or even stopped after 2 years? So you knock down the amyloid plaques and then you give time off, sort of like you might do with cancer therapy. Could we combine amyloid antibody therapy like Leqembi with another treatment? And would that be more effective? And these are the names of the other treatments. There's something called GSMs, which is very promising. Hoffman LaRoche has a big trial underway that are like a statin for Alzheimer's, something that would be a pill, not an antibody injection. And anti-tau, anti-sense, I mentioned that's something that Biogen is doing now. Anti-inflammation would be another thing. And then finally, what about vaccinating all of us against Alzheimer's? So that's quite remarkable, and it's underway. Could you actually give a vaccine that would be safe and effective and not as costly as giving people antibody infusions into vein every 2 or 4 weeks? And that's lying just ahead. So the bottom line is that Alzheimer's has been a big enigma. It's been very hard to figure out, but 40 years of science around the globe, in labs, in clinics, has led to this idea that we have now 2 drugs approved by many regulatory agencies that we can safely give to patients. They have some side effects, we can talk about that, but by and large, they're quite safe. So I'm delighted to take questions from you about all of this, but especially about things I haven't mentioned that you were hoping I would. And I hope there'll be time for that right now. Yeah. So we got questions already. Good. So we're going right around. We're trying to get them all.
On first? Okay. Who's on first? Right?
You.
It's on second.
I'm going to leave this up as a reminder of what the summary says. Yeah.
There's a center, they call it the Memory Center at Penn Medicine. One of our friends hoped to have, I guess, this kind of treatment because that's becoming more standard. And he was told that some bleeding that was noticed in his brain made this too risky.
Mm-hmm.
Is that right? You don't think?
I think it's not right, but Penn is a terrific place. You're talking about the Penn Center. Right. So I don't think that's the right way to phrase it to a patient and a family. I do this all the time. We meet with families, we tell them the risks. And it's true that Leqembi, the first approved drug, which we've given to about 800 people, has a risk of microhemorrhage. Micro means less than 10 millimeters in diameter. Not big enough to cause a stroke or cause symptoms, you notice, but you can see it in the brain on MRIs. Therefore, we get MRIs before the second dose, the third dose, the fourth dose, the seventh dose, and the 14th dose of this medicine we're going to recommend. And we've now just submitted our data for publication, and it turns out that less than 5% of all the patients we treated, now 850 or 900, had any side effect of that sort that they noticed. So Alzheimer's is a terrible disease, and not treating it means it just advances. These are the only treatments approved in the world for Alzheimer's. We think that it's worth the risk, but we ask our patients to hear our statistics and make their best judgment. Do they want this or not? There is a risk of microhemorrhage. Almost never is it a big bleed, like you hear people having a stroke with an explosive hemorrhage in the brain. But even that has happened in 3 or 4 patients out of thousands treated. So we don't want that to happen to our patients. Therefore, we carefully test them before they go on this. 4.8% of patients have any symptomatic side effect from this treatment. And even those, we stop it if we get bad symptoms, and it clears away. Yes?
Well, how do you measure the amyloid?
Mm-hmm.
And I have another question
Sure.
related to amyloid.
Right, so I'm going to show you again how we measure it. This is the way, this image, we have a remarkable diagnostic test called a PET scan, not PETs at home, but positron emission tomography.
Did it for cancer.
Yes, and PET scans used for cancer, for sarcoidosis, for many different disorders, rheumatoid arthritis, etc. And the PET scan is with a special chemical we put into a vein that goes to the brain and binds to the amyloid deposits. Now, it doesn't clear them right away, it just marks them for about an hour. And through the wonders of engineering, we can see the image. And that's what you're seeing here. The red color is that PET ligand or PET chemical.
Now, what is the chemical bond with?
The chemical is binding to the plaques in the patient's brain.
I know
And
that, but what is the chemical?
it's Pittsburgh compound B in this image. It has a chemical structure. You can't buy it off the shelf. It's something we have. It's all hospitals around the world. It's called Pittsburgh compound B. What's that?
Does it have a metal in it?
No. No metal.
That's my next question. But
Yeah, no metal. Yeah, we know that there's a relationship between metal ions and Alzheimer's, but we don't give any metal ions. So the answer is no, it doesn't have a metal ion. And by the way, this scan, it washes out of the brain in about an hour and doesn't cause any side effects. So this imaging that you see here in red is the amyloid plaques in the patient's brain. And the test is harmless. I'd have my dear wife, Polly, who I do love, have this test in a moment in order to find out if she's eligible for this treatment.
now the other question is related to metal. My sister had Alzheimer's, and there's no one that has ever had Alzheimer's. But she had rheumatoid arthritis and had many bouts of gold therapy. And I wondered if that had anything to do.
So I can tell you honestly, having studied this disease intensively for now 50 years and aging myself here, we've never found clear-cut evidence that metal ions, and certainly gold or other elements of that sort, cause Alzheimer's disease. They might accumulate alongside this badness, these plaques and tangles, but we don't think. And many of you may be old enough to know that there was a theory that aluminum could cause Alzheimer's disease. That's when I got into the field 50 years ago, and it didn't pan out, to make a bad pun. So it didn't work out. Let me take some other questions. Yep, go ahead.
Dr. Selkoe, question.
Can you tell us in layman terms, please, what are the red flags we should look at that show normal memory loss and Alzheimer's?
Right. So the red flags for Alzheimer's versus normal memory loss are little everyday events, like the name of an actor in a movie you saw, or the name of a book or novel that you read 2 weeks ago. If that doesn't come right up, that's what we call benign senescent forgetfulness. I know I have it, and it's okay. It's not a sign. If you forget that you ever went to the movies with your husband or spouse, then, and you don't remember that at all, that's an episode in your life that you've forgotten, and we call that episodic memory loss. So if there's an important thing that everyone in the room would say, oh, you should remember that you went to the movie. I don't care if you don't remember that it was Meryl Streep who was the actress. You can come.
Particular movie or all?
All movies.
enough for example.
Okay, so I don't know why you asked one particular movie.
It's one thing to forget that you went to a particular movie, but if you've never remembered never going to them.
No, if you forgot to go to any movie, any particular movie, because you don't remember going there with a friend or a spouse, then that's of concern. If you forget having been at this event, and if we see each other on the street in Palm Beach tomorrow, and I said, hi, I think I saw you, you asked a very good question, you said, who are you? I don't know you. You're not a friend of my family's. So that kind of missing whole important information, not remembering if you're so blessed your dozen grandchildren, and having trouble connecting the 12 of them to their 4 parents, making that connection. So forgetting important things, that's where you have to go to the doctor and find out. And if you're not sure, go to a cognitive neurologist or a geriatric psychiatrist and find out, does your memory, we give you memory tests when you come to see us. No one gets on these medicines unless they can answer these questions. I'm going to give you 3 words, an address, 479 Boylston Street, a blue Toyota, and a lampshade. And I'll actually test you guys right now. Try to remember what I just said, and I'll ask you in a few minutes what that was. And of course, when you're one-on-one with me in my examining room, you're going to make your best effort to remember 479 Boylston Street, a blue Toyota, and a lampshade. They don't mean anything. That's the kind of memory testing we do. Let me take another question.
Right here.
Dr. Selkoe, my understanding, and correct me first of all if that's wrong, is that there are some people who have a fair amount of Alzheimer's beta in the brain and do not develop clinical Alzheimer's disease.
Right.
In your opinion, is it that they will eventually develop Alzheimer's, or is there something else going on in those people which explains it?
Right. We have some very good evidence now that people who have quite a lot of this amyloid buildup in their brain, but they're still talking a good game. They're still able to make change at the checkout counter. They are highly likely to develop Alzheimer's later on. And it's like in your field, Ken, if I have bad-looking coronary arteries, atherosclerotic disease, I might never have had a twinge of angina or had an actual heart attack. But I'm at risk, and my cardiologist is going to say, you know, your arteries don't look good. Let's put a stent in. Let's make sure you're on a statin. You should have been years ago. So we think that this is pre-symptomatic Alzheimer's disease. Now, not in every case. There's some people who live to 85, 90, 95 and have quite a lot of amyloid. They're like the people in your practice that have quite bad-looking coronary artery disease structurally in there, and they never actually had a major medical event, a major setback like a heart attack. So we know that there's a variety of responses to a pathologic process. And we now know some of the genes that allow a person to build up amyloid and not actually get forgetful, which is wonderful. We want to see if those genes, which we know the names of some of them, could be amplified in other people.
I go to Publix in the morning. I have written down a couple of things that I'm going for and put it in my pocket. I get to Publix, I can't remember what pocket, I end up buying a couple of other things that I now remember I should have, that I need. I pay up and I leave. Then I get outside and I fish in my pocket and I find the things that I actually went there for. So I get those. But
That's an example, in my opinion, of benign senescent forgetfulness. It's something that many of us have, get a little confused, and it doesn't automatically mean Alzheimer's disease, because you eventually find it.
well, the other thing though is, I have a friend of mine that I was in college with 70 years ago. He comes into Greens and he says, yeah, you remember when we went down to where, I remember exactly.
Mhm.
But if you said, what did I have for breakfast? Who knows?
Yep. Yeah, that's very common. We can't remember things that just happened, especially if they're not terribly important. We've not been trained throughout life to remember exactly what we had for breakfast this morning. So that kind of thing could be a first symptom of Alzheimer's, and the only way to settle it is to go to a specialist who knows about this. But by and large, little minor things like what I had for breakfast don't matter.
Well, the other thing is, I take Eliquis, so this is something I couldn't.
Yeah, you would not be able to get on this particular medicine, but there's a new one coming down the pike you can take Eliquis, which is great. So that's not yet approved.
Yes, sir. What do you recommend for senior forgetfulness? Is there a drug or something that will help us remember better?
Yeah, there really isn't a medically proven treatment that helps everyday minor memory difficulty. I wish I could tell you one that does. What we say is staying generally healthy, doing exercise, having good oxidative metabolism in your body, avoiding head trauma, so wear your bicycle helmets, etc., and taking it slow. When you get a bit older, your pace of doing things may slow a little bit, and you have to respect that. So if you have a calm, quiet life, if you exercise regularly, if you have the Mediterranean diet, many doctors will tell you that's a benefit. So you avoid some of the typical American diet. I had a cheeseburger yesterday, so I'm ashamed to say that. The reality is that you can help your minor memory difficulty, and then you have to choose your parents very carefully, because if they didn't have any sign of dementia or Alzheimer's late in life, then you're more likely to get better. But it's really exercise, diet, avoiding head trauma, leading a healthy life. Many of us who do that will make it well into our 80s and maybe into our 90s without getting Alzheimer's. Yes?
What, the infusion, it appears that that's for stage 1 only. Is that correct?
I'll explain. So the treatment I talked about, the one that looks like this, that removes the amyloid like this, and on the right is the removal of amyloid, that is for what's called mild cognitive impairment, or MCI is the term we use, which is the very first sign of an Alzheimer process. And then the next step is called mild Alzheimer's disease. So we have mild cognitive impairment, then mild Alzheimer's. Those are the two early forms. Then comes moderate Alzheimer's, then comes severe Alzheimer's. We don't treat any patients with these new agents who have moderate or severe, but we do treat MCI, mild cognitive impairment, and mild AD. And we have cognitive tests we give you where we can score you and find out, are you still in the MCI phase or in the mild AD? But either case, you can get it. So this is for the early phases of Alzheimer's.
So what can you do, if anything, for the second phase?
So the second phase would be moderate Alzheimer's disease, or the third phase, really, MCI. Then there are 6 phases altogether, because there's something having what Ken referred to, amyloid buildup in the brain, but you have no symptoms at all. That's the very first phase. And if you have the third phase, which is moderate Alzheimer's disease, then these treatments are not likely to be beneficial. They might help a little bit, but we don't offer them. And there isn't a lot you can do. When you move beyond mild Alzheimer's, the truth of the matter is that anybody who says they can help you with that, they don't have evidence on their side. And we want to catch it early. So the amazing thing about prevention is there's a big trial underway around the world of this same drug Leqembi that I've been touting here. I don't have any stock in the company, so it's not for financial reasons. And that is being used by my colleague Risa Sperling at the Brigham and elsewhere to prevent the beginning of symptoms before MCI, before mild cognitive impairment, when we're like we think we are today. But we have plenty of buildup of that amyloid protein that we can see on amyloid PET scans. That's pre-symptomatic. That's like treating your heart disease before you had any angina, any heart attack. That's when your doctor wants to treat you. And we're going to find out in late 2027, early 2028, if this trial that Risa and her colleagues are doing called AHEAD, you can Google AHEAD on the computer, and you'll read all about this great trial. They have 1,300 people in it. They are perfect on their mental status test. They do remember the 3 items that, you remember what I said?
Yes, I got it. I'll give them to you backwards. The lampshade, well, the first one was the blue Toyota, second thing Toyota, and 479, I guess, is where you work.
Yeah, Boylston Street. It's not where I work, but absolutely right. So he's doing good. That perfect score you get on these kinds of tests, that's what you have to have, a good, perfect score to get into Risa Sperling's trial that's called AHEAD. And AHEAD uses this same medicine, Leqembi, to try to prevent you from getting Alzheimer's in the first place.
Why do you only treat 800? There's 800,000 that people should.
Well, in order to afford it, so we treat 1,300. No, because the companies and the cost of doing all these PET scans and following the MRI to make sure you don't have side effects and watching out for that bleeding.
You said you treated 800.
Oh, we've treated 850, but they already have symptoms. That's in practice. There are probably thousands and thousands here in Florida.
What you're doing.
Yes, doing what I'm doing as a regular practitioner. But I offer my patients who don't have any memory difficulty, who are all right, I get them to have a scan. If they have amyloid, but they're normal cognitively, then they can go into that trial. And the reason we have 1,300 is because there's just so many we can treat. Eventually, if that is successful, everybody in this room will want to line up to get that treatment. Yes?
The treatment itself, the initial treatment you're talking about. So you give that now over a period, let's say, 10 months or something like that.
About 2 years.
2 years, really. So people get these treatments every couple of months or something.
They get them at the beginning, Leqembi, every 2 weeks.
2 weeks.
They go to the hospital. But a big breakthrough from the FDA a few weeks ago, you can now take it by auto-injector at home, the way that we take GLP-1 agonists for weight and the way we take insulin. So now you can start at home.
No side effects?
The side effects are what I mentioned earlier to this gentleman, that about 5% of patients can get symptoms. Right.
And how successful? How long has this trial been going on?
So what I'm describing now in people who already have symptoms is no longer a trial. That trial was quite successful. We're now just following patients who've been given the treatment in everyday life. It's not a trial anymore. Anybody who has memory difficulty can get on this drug Leqembi. The prevention trial I talked about, where, like this gentleman, he got all the answers right, that we don't know how that's going to work yet. We're waiting to see.
But you see over time that they lose
Yeah.
this plaque condition.
Right. They still decline. The patients still decline. That's shown in these graphs.
So if they don't have the plaque, why are they still?
They do have the plaque. Anything that I've said today in this forum is people who have the plaque already. Either they have the plaque and they don't have symptoms yet, or they have the plaque and they have symptoms.
This treatment is supposedly getting rid of the plaque.
Right.
And
And
if it reaches a stage where,
Yeah.
where you show no plaque, what does that mean to the person?
what it means to the person is that they have less likelihood of advancing or progressing, but not zero likelihood. If we treat them very early in their plaque buildup, when they have quite good memory, but just a little bit off, then if we block the plaques and clear them, they tend to not get worse. So we have data that the mildest patients in our trials, followed for 36 months on this agent Leqembi, don't deteriorate hardly at all.
And it doesn't come back to plaque.
It comes back if you stop the medicine, but only very slowly. 2 to 4% of the load that you get at the beginning comes back per year. So 2 or 4% amyloid buildup per year. So we can stop it eventually, but truthfully speaking, I'm going to recommend that people come off of it for 6 months, then go on it again for 3 to 6 months, then come off of it like other treatments.
So, listen, we all get EKGs every year.
Mm-hmm.
Get ex-cancer patients, get PET scans 6
Right.
months. Should we get brain scans once a year?
I
6
think
months?
the answer is we have something better for you than a brain scan. This scan that I showed you earlier, that's the PET scan, this thing.
Was it the same
But we have a blood test now. I didn't tell you that. See, I have a lot of tricks up my sleeve. So the blood tests can be done every 6 months or every year, and they give an indication, are you building up that amyloid protein in your brain?
What's that called?
It's called PT217. PT217. It's phospho-tau.
Remember everything.
Better to write it down.
2-1.
And that's being used in doctors' offices in Florida and around the country and around the world. So it looks very good. I think I better stop or I'm going to get the hook here, but I'll stay around and ask another question.
Good call.